Okay. Good evening once again, everyone, and good morning. Am I audible enough? So today we want to discuss this article, and is titled Furin-Mediated Intracellular Self-Assembled Olsa-RVRR Nanoparticles for Enhanced Magnetic Resonance Imaging and Tumor Therapy. Okay, why did I select this article? Number one, it's a Nature Materials talking out. Most articles published in Nature Materials talks about new innovations of materials used for diagnosis or for therapy, and for some other parts of science. And also with a good impact factor, with 38.8. And also because of CEST imaging method is used in this article. CEST imaging method means chemical exchange saturation transfer method. It's an imaging method in MRI, which is becoming more emerging day by day. And also because it's a tumor-specific intracellular assembly for tumor imaging. Like recently in research, we discovered that research is going to be passed into personalized medicine. And this is one of the way of having a personalized medicine. And also the designs and the experimental design and methods are very explicit enough, and anyone that pick up the article can easily follow the experiment to have a turn. And about the author is Yu Yuan, and the correspondent author is Buffett GFWM. Reason why I'm stressing this is because the author is one of the major in CEST imaging all over the world, and is also from Johns Hopkins School of Medicine, and they've really done a lot of work on CEST imaging since its existence. And when it comes to CEST imaging, these are the relevant institutions: Johns Hopkins, Vanderbilt University, Johns Hopkins, Kennedy, and a lot more, even talks of German and the likes. Yeah, this talk still talks about the Hopkins. These articles they've published under CEST imaging. And this author is mostly one of the corresponding author. And for CEST imaging, for those of us that have been interested, over the years, this is the plot of what has happened over the years in CEST imaging, as we can see, and they are still publishing articles. Yeah. Now, for us that might be thinking, is this really relevant to medical physics? Yeah, this is, this is the most relevant authors when it comes to chemical exchange imaging in all over the world. And this particular person this is a medical physicist in Germany. You can see this is the fifth relevant author in this field. That's even one of these things that really raised my open. I started research on it, and I saw that this man is also relevant in this. He's a medical physicist, and in all of that article, we boldly write it there. I'm a medical physicist, and I also discovered that when it comes to the simulation aspect of this field, because it requires some simulation, it requires some analysis, computation analysis. This is always done by him. So most authors usually collaborate with him, because being a physicist, they need to collaborate with him to get their results. Even after the radiologists have they've gotten their images, they still need to collaborate with him to bring out the beauty out of the work. Chemists also collaborate with him. No matter the number of chemicals they've designed, they still need to collaborate with him to bring out the effects of what they have done. Further in the you will understand better why I said so. So let's quickly go to the background. I want to just quickly give a background of what is chemical exchange saturation transfer imaging. It's just talking about exchange with water protons. And just like this, you see, these are the green ones are the water, are the CEST agent mobile protons, and this is water molecules. And this KEX means an exchange rate. So what happens is that if an RF is applied on this, on this particular, let's say this is a phantom, or this is an in vivo environment. If an RF is applied at a particular frequency, it causes an exchange between these protons with these other protons. And when there is an exchange, it shows an effect. The signal effect, there is a signal effect at this particular exchange point, and that is why they come about the word chemical exchange saturation transfer. And also another thing we should want you to notice that these agents, sometimes they are endogenous agents, like creatine, agents that are found in the body, creatine, Latin. These are agents that they are already in the body. They just go into the body and image it. And also, just like in clinicals too, we have the APT imaging, which is the emit proton imaging. They don't need to give any contrast agent. That's the beauty of it, before they perform an MRI using this method. And this is the formula calculating. This is the water exchange without RF, and this is the CEST exchange. And also, for every exchange to occur, this principle must be obeyed. The change in exchange must be greater than the exchange rate of the molecule. So if this is not greater, then no exchange will occur. And that's why we have that. You can see this peak. This peak shows at this particular point there will be an exchange. And it's always, to get this peak, is always for us to subtract what is the signal intensity at this point from this point. They will subtract it together, then we can get a change in W, which is the CEST effect of that particular phantom made. And also this still explain further the MRI CEST experiment. You can see an RF is being applied, an exchange occur between the bulk water and this CEST water, and it gives this at this particular point. And we can see what happens. So at this point, that we calculate this and we say, oh, there is an exchange at this point. What is the saturation percentage at this point? All this still boils down to physics. Now, see, the net saturation percentage effect is calculated as 1 minus IS. IS is the signal intensity at this point, divided by the signal intensity at this point. It's covered at this point. We say that there is no exchange at all. We can see when the signal intensity is up, we say there is no exchange. So we subtract from that exchange, multiply by 100 to give us the signal intensity percentage effect. And all this calculation these simulations, this idea was being brought about from the man I discussed earlier, Morris. That it was one that was able to get this method out for people to calculate the ST percentage. So showing the effects we can have as a medical physicist. And this is also talking about the sensitivity, how sensitive these CEST agents are. They are they are sensitive. And if we have a good environment where there's a good, not a large number of exchange, then we can get a good signal intensity. And also, in terms of simulation, this is the equation for simulation. See, this simple equation extend into this for simulation, and also to calculate the exchange rate. You can read further on that if we are really interested in it. And you can see these are the simulation parameters. And also, this is also the mechanism. See. Okay, another way to also do this is to also calculate, make this the I naught. The I naught means when we apply the RF and there was no exchange, with this exchange, then with that we can get the exchange rate. So let's quickly go down to the business of the day. So now we are talking about intracellular self-assembled nanoparticle. So why intracellular? Intracellular self-assembled of molecules, drugs into nanostructures is among the effective strategies used to increase the concentration of drugs locally and to prolong their exposure time. And also, it increases the local concentration of anti-tumor agents. And increase sensitivity while minimizing non-target toxicity. Because if it's self-assembly, it's just going to be focused based on where the area it's been targeted in the body. And also for this article, they used a collateral cancer, and which Yemi has really stressed on in our first seminar. So if we are interested, we can go to our YouTube page and look at the go through the first webinar, which really expatiates on why collateral cancer, why it can be a focus in Africa, though it doesn't. The level of resistance in Africa is a little bit low, but it's also an important cancer that needs to be considered. And also, the potential benefit of tumor-specific intracellular assembly prompted the development of an enzyme-responsive theranostic platform for tumor imaging and therapy, and that's why they used an enzyme. And what is the enzyme they use? They use furin. Furin is an enzyme, and is a protein convertase upregulated in multiple malignancies. Also, it's a potential therapeutic target and a biomarker for prediction of tumor progression. And it's also want you to note that it has a degree of expression that correlates to 5 years survival rate, which also talks about the survival rate of a cancer patient. And why olsalazine? Olsalazine is a small molecule drug, which is a DNA methylation inhibitor. It has potential broad spectrum anti-cancer agents. It also belongs to a group of silicates. Silicates such as the polylysine. Silicates are known like aspirin. So this drug belongs to a group of silicates. Aspirin is a silicate too. And also, the hydroxyl proton on this provides a distinct contrast for it to be a chemical exchange agent. So, and also, let us know that for us to use an agent or any substance as a CEST agent, it must have a proton or an exchangeable proton on it. Just like research has been done using glucose, using lactose, using sucrose, using glycine, because they have exchangeable proton. Even glucosamine has also been used as CEST agents. If people are just developing every day, can just look and say, this substance, does it have a proton? You can go to the lab and test it and see if it can work as an imaging agent. And this is the schematic structure of how this drug is being produced. I might not bother us with that, but we studied very well, talking about how they fused olsalazine with RVRR, which is a peptide, to bond it together. And with that, after then, they fused a furin. After fusing a furin, they can get an olsalazine nanoparticle, and this shows how it works in the body. How it works, leaving the Golgi part, going to the membrane, passing through the cell membrane, and this is the Golgi apparatus, how it works in the body. And this shows the nanoparticle infused with furin agents. And don't forget, the furin is an enzyme. So fused with the furin agent, then applying CEST on this nanoparticle, then it has an effect, inhibits the DNA in this particular point, and be able to do simultaneously undergo a tumor therapy and also an imaging simultaneously. And this just explain briefly the production, which is also an anti-cancer agent was conjugated to the cell-penetrating peptide RVRR. Then after then, a CEST imaging yet, which, like I said, anti-tumor drug activity, and also it induces penetration of the agent across the membrane, and that as a substrate cleavage enzyme furinase. Please, Yemi, please, you need to notify me if my time is going. Thank you. And what is the study hypothesis is to say that the catalyzed intracellular formation of olsalazine nanoparticles may not only enhance the intracellular concentration of olsalazine, but also prolong its retention time, leading to an amplified localized olsa-CEST signal, as well prolonged drug exposure. And this shows the flow chart of the experiment. The methods they undergo in vivo in vitro CEST experiment. In vitro means not done on animal, done on maybe production of phantoms and some things to characterize the probe, to characterize the nanoparticle probe, to see what it's all about, and then they now go into the in vivo studies and the in vivo cell studies. And that's why I mentioned earlier on that the in vivo cell study was done on breast cancer. And they tested about 3 cancer cells, and they discovered that the HCT116 was the most effective. We see this further in our results, and that was also the same collateral cancer, human collateral cancer cell they used also in the paper Yomi presented earlier on. And the in vitro cell studies, they undergo a GSH detection, a cell uptake, efflux, and viability studies to see what is the uptake level of this particular nanoparticle. And they did an HPLC analysis, a live cell imaging of cell assembled nanoparticle, and they also do a 3D SIM super resolution microscopy and image analysis and NIRAMA. All these experiments, if you have gone through the article, has reasons why they've done all these experiments to make it, to validate the toxicity level, to validate the availability, to validate the uptake level. And okay, let's briefly, I'm just going to show a few results, and this is just showing the physiochemical characterization of the OLS-RVRR and the Olsa nanoparticle in solution. Okay, you can see this is the ordinary Olsa-RVRR, and this is the Olsa plus GHS and plus furin. And you can see here, you can see that virtually on this line, really no signal. But if we can look at this critically, you can see that a little signal was here, and also the point at which it exchanged is different from various. Looking at the time, is very, very different. And also this shows the conversion of the probe of this probe with time. You can see from 0%, looking at, and this also shows the retention time. This also shows the retention time of the probe involved, of the nanoparticle probe. So it's showing the enzymatic conversion of this with time. And this is the TM to show us the size of the particle. This is to show us the size of the particle, and this will give us, this is also the DLS to show us the size, and also what happens after incubation. What did they get? So this will tell us this. And this is the my business of the day in this article. This shows the saturation effect. Now the probe was scanned at various B1 power, which from 1.2 to 10.8. Mostly we do this in our experiments to show which one has the best effective power. Then from there we can now choose one power further experiments. We can do that for further experiments. And this shows the empty asymmetry, of the probe. See, because for every saturation transfer, for every saturation percentage, there will almost be an empty asymmetry, and this is gotten by changing W over I naught over the scan, the point at which there was no exchange, the initial points of scanning. Okay, so now let's this is also, let's go through the next. So now this shows the empty asymmetry against the saturation transfer feed strength, and with that you can see at an R square value of 0.99, which an exchange rate of 697 at. And also, so you can see comparing the three agents at 3.6 microtesla, you can see what they got here. See what they are comparing. So show the Z spectra of the and from there we can see which one is more effective. Although both of them are exchanging at the same point. They're exchanging at this ppm, it's around 9 ppm, but at least it also shows the effectiveness of when they added maybe no effect of when they added furin. But at least the percentage is also a bit more better at this when they infused GHS plus furin together. And this also shows the empty asymmetry at B1 equals to 3.6 microtesla. And this is a computational work that is being done in MATLAB to show the fitting of with concentration, empty asymmetry concentration against to see what is the effect with concentration of this nanoparticle. So for various nanoparticle was produced at various concentration and was scanned, and we can see the plot that the empty asymmetry increases with concentration, showing that it means that this particle is concentration dependent. And also we have the empty asymmetry also with pH. So what is the effect of this particle with pH? And we can see it also increases, the empty asymmetry percentage also increases with increased in pH. Okay, my time is going. So I'll just quickly rush through this. And so also this talks about the cell viability, and this talk about what is the drug efflux of this particle. And this is the in vitro study, cell study. And if you have gone to the article, we would have seen the in vitro cell study. They also compared both, and this is where they compare the cells line, and you can see the cell line that is being fused with HCT, the colorectal cancer, really gives a good effect. And this is the in vivo study, they study on xenograft, and then both cells were planted at various sites. And if we look at the trend at 2 hours, there is an higher signal intensity for HCT116 than LoVo cancer cells. And this also shows the empty asymmetry of the cancer cells they compare. And this shows the effect on the essential organs on the body, which makes it a little okay. And we can see also the signal intensity is very low. So it means that the nanoparticles are not retained on the liver, and they are not retained on the lungs. Okay, so it shows us that the pH independence of this Olsa-RVRR signal will enable the use of Olsa in different furin expressing tumor models without contaminating signal changes associated with endogenous tumor derived pH changes, and also the enhancement of acquired Olsa signal for the furin expressing HCT116 tumor xenograft is about 4.3%, which is higher compared to other CEST detectable compounds, that is 1% for dextran, 2% for glycosylated mucin 1% for the reporter probe, and 5-methyl, 5,6 dihydrothymine, and these are the CEST agents they've used. And you can see that this CEST, this Olsa CEST has far more better exchange rate. Saturation percentage is at 4.3. Even for hydrogel, injectable hydrogels, it's between 2.3 to 3%. So what is the conclusion? You can see that they were able to successfully develop the Olsa-RVRR, and also it also shows the efficacy of this nanoparticle for inhibiting development of colorectal cancer in preclinical models. And also being used as a therapeutic drugs make it with an inherent CEST MRI contrast agent with no additional labeling of molecules, makes it a potential drugs for potential drugs and also for imaging. And unlike the CEST signal for salicylic acid, where the proton exchange rate is stronger dependent on pH, this is not dependent on pH. And also the signal is stable within the pH value 6.5 to 8.0. So what do we learn? We can apply in this new application technology of intracellular self-assembly. We can apply it to our work, and also we can also focus on development of new nanoparticle, which is tumor-specific enzymatic reaction. We have a lot of enzymes there, apart from furin, that has been discovered to be maybe for drug therapy. We can look into that and see, and also we can also explore a new recently developed approved FDA drug, silicate small molecule drug, which has the possibility of being a CEST agent for our imaging. And also another thing I also draw from this is that in my course of study, one thing that's been coming to mind is that we have a lot of plants in Africa that we can also look at and explore. These plants contain some polyphenolic acids, and they contain some, just like our regular bitter leaves. So if we can look into this and see if they are, and this some in articles we've seen that, like I was just reading about bitter leaf yesterday, that was saying they've seen it as an anti-tumor. It can be used as an anti-tumor agent. So if we can explore more of this to work as a therapy, and if it also has an exchangeable part, it can also work as an imaging agent. And also, as physicists, we can more work are still being done in simulations and modeling to improve diagnosis and also to improve final results based on our treatment method back home. Thank you for your attention. Hello.
Thank you very much, Mrs. Oluwatosin, for the wonderful presentation. I think I really learned a lot from the presentation about CEST imaging and also about the research. Once again, thank you very much. We are going to open the floor to everyone to ask questions and to and help with the discussion, just to facilitate the discussion. Right from the presentation, what's our reaction? What did we pick from the paper when we were reading it? And hopefully we think it's something we need to discuss about. So I'll leave the floor open. Please indicate once you have a question so that we can move on from there. Thank you. Okay, I do have questions, and maybe we'll probably start from mine, which is fine. And also I encourage anyone that have questions to please indicate once they have one. So I started by looking at DNA methylation and why it was specifically the point of focus in terms of using an agent that is a DNA methylation inhibitor. So I want to find out if in that regard, just more explanation in terms of DNA methylation, because in my thinking, I'm thinking it can work both ways. Are there situations where we don't need to inhibit the methylation process? That's what I'm thinking in my mind. I don't know if my question, if you get my question clearly.
Yes, I got a question. Actually, it's not as if the DNA mutation is their focus there, but when they were choosing the drug, you know, for every drug you choose or for any therapy you effect, there must always be a biomarker. There must also be what you are looking at. Maybe you're looking at an EGFR inhibitor, you're looking at other type of, maybe is it TK inhibitor now in the body. So if we are looking at, assuming they are looking at a EGFR inhibitor, they will not choose olsalazine because olsalazine might not be an EGFR inhibitor. And looking at the characteristics of the cancer they used, obviously It must have been one that inhibits DNA methylation. Did you get that? So
Yes.
with that, they need to, so for every drug or for any element we used in therapy, we must consider what is the biomarker we are considering, what is the characteristics of the tumor we are considering, and what is the characteristics of the drugs we are considering. So
Yeah.
hope your question is answered.
Yes, yes. Thank you very much. Any other person with questions?
Hello, can you hear me?
I can hear you.
Okay, I just want to say, firstly, I mean, that was an excellent presentation, Professor Tosin. Hello, can you hear me?
Yes, yes, we can hear you.
Oh, okay. So I just want to say that was an excellent presentation. I mean, I'm just thinking about so many things, and I don't even know how to put my thoughts together. But sorry, there was something I did not understand. So the imaging. So how exactly is this imaging done? When you mentioned it's in vitro. So these tumor cells are injected with these nanoparticles, and then an RF signal is sent into the in vitro sample in the dish. Is that it, or it is passed through? How exactly? Okay.
For the in vitro,
Okay.
for the cell imaging, what they do is to put the cell together with the freeze together with nanoparticle. Then they image it also on an MRI machine, like a phantom.
Okay. So
For
okay.
the
So it is.
experiment.
Okay. So is it a standard human MRI machine, or do you have like
No,
what?
like for this for these experiments, they would have used an MRI machine, a small animal, because they used rats. So they would have used a small animal MRI machine.
Okay.
Since it is a like just like in my lab, I normally use a small animal MRI.
Oh, okay. Okay. Then I'm also just thinking aloud. I mean, thank God you mentioned theranostics. So this has potential, while you're imaging, you can treat at the same time.
Yes.
So I'm just trying to
That's
see
why they use the
how
drugs.
Yeah. So I'm just trying to think about how it compares to nuclear medicine procedures. Like, I don't know if you've ever thought about which is it better? Is it more beneficial? Is this better? Is this cheaper? Is this more specific? Does it have a high specificity and a high sensitivity compared to, using radiopharmaceuticals in nuclear? I don't know if my question makes sense.
I understand you. Just for us to compare.
It would be nice to actually make a comparison.
Yes. I think
it would be actually nice to make a comparison between. Yeah.
I think from my study so far on this aspect of imaging and therapy, I can say number one is safer than nuclear medicine because it doesn't involve radiation. And as we know that MRI is safer, so it's safer. And for the sensitivity, they still have a little challenge of sensitivity, especially for those ones that you don't even need, because there's some imaging that you don't even need you don't need, what is it called, contrast agents for imaging.
Yes.
Just like the ones they do in clinic currently in using CEST, they use APT, which is the amide proton transfer. They just image the amide in the brain, which are mostly for brain imaging, and the sensitivity is low because no contrast agent was given. They just image based on the abundance of amide in the brain, which can exchange with the water protons in the brain to which they get an image. But if this, like also they've also tried glucose in clinics, because our body also abundance of glucose, so sometimes they fuse glucose and they get a good sensitivity, and we just need to work furthermore to increase this specificity and sensitivity. But it is safer than, I can categorically say that it is safer than nuclear medicine. And now with this article, they're already looking at personalized medicine, because it's self-assembled. It's not based on any chemical. It doesn't require any extra metals, which also have their own effect on the body. These are just drugs that are already been approved with an enzyme that is abundant and being fused together.
Oh, thank you very much.
You're welcome.
Thank you very much for the question. Do we have other questions from the participants, please? If I may, not much of a question though, but because while reading through the paper, I'm still trying to understand some aspect of it in terms of, because I know they did multiple CEST measurements, some in the solution and in vitro, then in vivo. So there was at a point on page, if I'm looking at it correctly, on page, just a second, please. Not on the first page. Yes, on page 1378. Yeah, where you have the pictures showing all the pictures colored figure right below figure 2.
Is it on the supplementary information?
No, on the paper itself.
Okay.
On the paper itself, 1378. If you're looking at after figure 2. So they mentioned
the page after figure 2, right?
Yes, just right on that figure 2.
About the methods.
Yes, when they got to CEST property of Olsa, in that case, there was a caption that says that when they use 5 millimolar of Olsa with RVRR, that it has the same signal strength with 2.5 millimolar of Olsas that was not fused with RVRR. And then later on, indicating that it was a little bit twice lower, attributed to the decrease in the number of exchange OH protons by half compared to the one that is free. But later when they talk about it in vivo, it appears the reverse was the case, where you have the one with RVRR higher than the free. So I'm trying to go from the information they provided there when they look at the sample. They compared that ulcers with RVRR with ulcers and NPs, nanoparticles, and then with free one, and it appears that the free one performed better. But in vivo, it appears it was the reverse. I don't know if you noted that and if there is any reason for that.
Okay, I get what you're talking about on the when they talk about the CEST properties, right?
Yes.
Okay. Yeah, I think what they are doing here is just to compare the efficacy of generating the CEST signal between the Olsa-RVRR, and I'm sure this will also inform which millimolar would they use for in vivo, because before we can do an in vivo experiment, we must have done an in vitro experiment to certify which concentration gives us a better signal effect. Okay, so they must, like now you see one of the figure, you can see they worked with at various concentration. I showed it on supplementary information, and I also showed it on my slide. Where yes, so at that point they will show which of the best signal can we take, and also you also need to also consider the volume in the concentrations of nanoparticles. Is also important when it comes to tumor imaging, because at a particular concentration it can be more toxic in the body. So with all these things would have been put in place to determine which concentration we use at the in vivo level, and that will give a good signal. And also, one contributing factor to the in vivo experiment is that there can be how will I use this word now? There can be some molecules in the contributing molecules in the in vivo environment that can also contribute to the CEST exchange because it is in vivo.
Yeah.
So not necessarily only the nanoparticle that was injected into the body. There will be, because like now, if like that's why they said it doesn't affect pH. If it's a pH, like a salicylic acid that's like polylysine that has pH effects, it's going to. And also we know that cancer tumors are also acidic. So now imagine giving giving an agent that has pH sensitivity in the body. Obviously, the acidic environment of the tumor environment will influence the signal intensity of with the particle that is being used.
Okay.
So that's why.
Okay. Well, thank you very much. Will the signal not be separable in the sense that the frequency we use for that excitation will be peculiar to the particular molecule, the stuff we put in, because you're doing CEST and you want to do a form of pre-saturation first. So I think that's why they talk in terms of the parts per million. That gives the frequency range that we want to work with. Maybe that would help to attenuate signal from other competing stuff in vivo. I don't know if that makes sense.
Yes. At that point, what we do is that after CEST experiments, we'll after performing the experiment, like the in vitro, or there's something we call that we put into some codes, some written codes, which you can optimize for your work. And with that, we try to separate. We have some properties we call the MTC, which is the magnetic transfer concentrations. Also, we also have some NOE effect, because sometimes you might even get a peak on your scan, or as a CEST effect after scanning, and it might not even be CEST effect. There might be NOE effect, nuclear ulcer effect, or magnetic transfer effect. So firstly, we must first note, are these the real CEST effect after we apply the RF? Then with that, then we have some, like I said, some simulation has already been done to cater for this NOE effect and the MTC effect. When all these effects is being removed, then you can now be sure that, oh, it is truly a chemical exchange, not an NOE effect or an MTC effect. Then we can now go ahead with the experiment. But it's very, very important to separate the NOF. And also, we also have some corrections. We have the B0 corrections, which also has an effect, and the B1 corrections, which also has an effect. So we do all these corrections before we can assume and say, oh, my spectra, I'm sure they've done all this. They've done all this before they could come out with this final result to say, oh, the spectra looks like this. This is what they are getting. I don't know if that answers your question.
Yes, perfectly. Thank you very much.
You're welcome.
Okay. We can take one or two more questions before we wrap it up for today, or comments in general. That would help as well.
Hello, can you hear me?
Yes,
we can hear you.
Okay, so this is not a question. Like I said, I'm just I'm glad I was part of this particular meeting. So at the end of your presentation, Mrs. this is this is the question I always ask. How do we bring this home? And I'm glad you mentioned it in your last 2 slides. So for someone who has no idea or has no bearing and is very interested in going further with this study, knowing more about CEST, and you mentioned something about using local plants.
Yes.
Plants, that we could. Yes, I would like you to kind of build on that, if you don't mind, because that is one of the aims of this, one of the objectives rather of this journal club, is to find out, see what is going on elsewhere, and how we can implement this, you know, in low resource settings, if I may use that word correctly. Yeah. So I would like you to just build on that. Yeah.
Okay, thank you. Just like I said earlier on, mostly the articles that I've seen, that I've read on this particular CEST imaging, what the authors have done is just to look for a contrast agent around them, something that has an exchangeable proton, and go to the lab and test it and see if it has a sex defect, and can this thing be used for imaging. And so also back home, we can look at what works around us. Like there is a particular author that works on pH sensitivity, and what are the agents he used? He used already approved X-ray agents, the ipamidol, the iobital, he tested all the X-ray agents, contrast agents that are being used for X-ray imaging, that already approved, that people already, and he tried to use them, and he got a good pH sensitivity using ratiometric method to see, oh, this element can exchange. We can use these elements are already approved and for use in X-ray. Why don't you transfer these same elements to MRI, and without the effect of the radiation effect? And it's working, and I think they're already on the plan to transfer to clinicals. And also, just like I mentioned earlier on plants, we've heard about, they've used salicylic acid for CEST imaging, polylysine. Aspirin is a salicylic acid. These are things that they are regular experiments that are regularly available around us. People just pick it up and use it. They've used glucose, the normal glucose we take. I even did, when I started this experiment, I also used glucose, normal glucose that we drink, just put in the water, go into the MRI scan and scan, and truly there was an exchange. They've used MET 3.0, which is also an approved glucose type of glucose. So why I mentioned the plants is, I've been thinking, I've been researching, although because no work, so much work has not been done to show us what is my next stage in that research is to see what is the exchange properties in this particular plant. I mean, from their extracts, like bitter leaf. Now, bitter leaf has some flavonoids, which have been known to have anti-cancer effect. These things that might not have been taken further. They've used on animals. Some people have used on animals. Oh, it reduces the effects of the tumor, or the tumors went down. So can we go further and take this the extract from this bitter leaf and see, okay, if it has an exchangeable proton in water, okay, can we use it for both imaging in MRI and also use as a therapeutic agent? Eventually, at the end of the day, we would have ended up doing our own self-assembly, looking at, okay, what are we targeting in the body? Which enzyme are we targeting in the body? Which of these plants have this enzyme that we are targeting in the body? And how can we make use of them? So that's my point when I wrote that point. Because, okay, I mentioned polyphenolic acid. A lot of polyphenolic acid are in our plants, they're in the food we eat. Okay, which of them have been extracted out that we can say this is the chemical properties of this particular phenolic acid? Okay, what if it has an exchangeable proton? It can either be an OH proton, it can either be an NH2 amine proton. Okay, if this proton, can we take it to an NMR machine and look at what is the ppm? Does it have an exchange at a particular ppm? And does sensitivity high enough? Then you can go further and go into experiments. And if it passes the animal experiment, pass the statistics experiment, this same contrast agent that we've already assembled can be transferred to clinical, which makes diagnosis and tumor therapeutic diagnostic very more effective. And with that, we have added to knowledge based on what is in our environment. So many things can be explored. Moringa can be explored. Shower sharp. Okay, recently I was reading an article on shower sharp. Somebody posted shower sharp on a group that his father-in-law, somebody used it for prostate cancer, and it was well. And I took it upon myself to go online, and I was amazed when I saw a lot of articles already published on shower sharp having anti-cancer effects. And these are what people don't even know back in Africa. because this research were done by international people. And you know, Africa, we don't read. I was surprised when I saw about 5 articles. I was like, oh, so this soursop has an effect on cancer, and already clinically proven, I mean, already scientifically proven to have effect. And these are plants people have in their compounds. And people might not even take soursop, just go and take it, not knowing that it has an anti-cancer effect. And if we have these home remedies for cancer treatment, I think it also be and it also improve the cancer. It reduce the cancer effects in Africa, because we have a lot of these plants that they are not even exploited, and also based on our experimental effects, no equipment, nothing to go about it. It really is worrisome, because these are some things that it's because all this experiment is a multidisciplinary experiment. The chemists to be there, the pharmacologists to be there, doing their own part, the physicists to be doing their own part by doing the calculations and see what can we effect. Then the nanotechnologists also be there. So I think a lot of things can be explored.
Interesting.
Hello.
Hello.
Yeah, thank you. Thank you very much. I thought I was not going to say anything, but you have actually lifted my spirit. I just wish so many people had come to partake in this wonderful experience we are gathering here. I think this journal club, if the way I'm seeing it, is going to engender a lot of things, especially in Africa. We have been taking the lead in Nigeria. I want to believe so many people will enjoy it very soon from other parts of the world, continent, to make it really international journal club, which I believe is the aim. In fact, physics is becoming much more interesting. You can see physics making a incursion into so many areas, biology, chemistry, and I just wish we can have this synergy between, between so many fields. Chemists coming together, biologists, blah, 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 physics coming together to form something and add to the body of knowledge, not just adding to the body of knowledge, making something that will be very, very clinically relevant. Before you know it, one day we might just have somebody from this part of the world and becoming a Nobel laureate. I really want to appreciate you for the wonderful job done today. I actually learned a lot today. So I want to thank everyone. This journal club, we should just try and keep it up, no matter the challenges that come our way. I believe these are just testing time. Let's just keep the faith up and see what we can do to make it much more lively. I know maybe so many other people would have joined. Maybe if we had done kind of reminders, also reminders many times as possible. Maybe probably more people would have joined. I want to encourage all of us. Today's presentation was actually a great one. Thank you. Not just for me.
Thank you, sir, for the encouragement.
Thank you, sir.
Thank you. Okay. So moving forward, do we have other comments? I think it's about time we wrap up.
Yep.
Even though we started a little bit behind schedule, just like we noticed the number in attendance, so we decided to delay it a little bit, but hopefully subsequent meeting we'll have more persons joining in to learn, because this is really an enlightening section to learn and to improve ourselves as medical physicists. Well, all right, till next time, I guess this is where we have to drop the curtain for today. Thank you once again, I'll say Dr. Oluwatosin, and for the wonderful presentation. Oh, I should use the word prof. Thank you very much.
Neither of it. So I'm still my own Bulte.
Okay. Thank you. See you sometimes.
Yes,
Okay.
and the video